Hello, welcome. On your screen here, you'll see a picture of some fish. These, I believe, are zebrafish, and if you were to go fishing in any ocean or pond or river or stream and hunt for a zebrafish that was one of these colors, you would, I would say, almost certainly not find them. You can buy these in the store. I was at Walmart a couple months ago before the quarantine and happened upon these fish. They're called glowfish, and they are a bunch of nice, bright, different colors. Um, they are not. You're not going to find these in nature. These were um, modified in order for you to enjoy them as pets. They are have had a gene inserted into their genome that allows them to be these colors. And it is not just fish that have been modified in order for our enjoyment or otherwise, but also, if I can find the right one, a whole bunch of other animals have been. Do a Google search. Some of these are not. Most of these are real, but cat. You've got the cat that's glow in the dark. You've got this chicken. I tried to get this one up, but it wasn't clear. This is, I think it's either mice or rats, where they glow in the dark, and they've added a particular gene. Here's some rabbits that glow in the dark. Here's looks like some sort of monkey that glows in the dark. This is a picture I've seen many times before. You can see, and this is a some sort of weird-looking rooster. But these glow-in-the-dark particular ones have used the glow-in-the-dark fluorescent protein from jellyfish, which is one of the first isolated genes, and uses to modify organisms. You'll see a lot of mice and pictures and whatnot, but it's been expanded, with technology and what they can do, and now you've got all sorts of large mammals that have been modified to glow in the dark, or just be a color. I believe this sheep has been modified, and it's in the dark, and they're probably using some sort of light, and it's kind of reflecting or glowing in that light. But this all goes back to genetic engineering, and what we're trying to do, what scientists have done and the technology to do now, is to intentionally change the DNA of an organism. All right, so you're taking either a gene that is defective and you're trying to change it, which you would do with humans and genetic disorders, or you're adding a gene. Right? That's why with bacteria, with insulin, they added the insulin gene to bacteria. Now they make it, and they can get a useful product out of it a lot more effectively than you would by harvesting it from pig spleen. In the plant realm, you have plants that have been modified to resist insects and resist and grow better, have better fruit. And there's also other aspects of that, or to, if you have some companies like to make weed killer, and if you have a weed killer and you're making plants, you don't want the weed killer to destroy the same plant, so you put a gene in there that helps you stop from being destroyed. But that's genetic engineering. So for the next couple lessons, we're going to talk a little bit about this. I'm by no means an expert, and we are going to kind of cover kind of the margins. There's a lot you can do with this, and the technology for this is getting better and better every day. All right. Well, if you're a scientist and you're trying to edit a gene, the first thing you need to do is find a target gene. All right. So remember, when we're talking about genes, you start with the DNA, and that's what you're going to target, changing the DNA, or adding it, and then finding what it was, that protein, right? The DNA goes to RNA, and then you have a protein. It's the protein that you're interested in the ultimate outcome. Right? There's that green fluorescent protein that is making animals glow in the dark. But first, you need to find out what that DNA is. And to get the DNA, right, you can have all sorts of proteins that you're looking for, but ultimately you want to find the DNA. You can isolate and look for and try to kind of sequence the DNA in a lab, which is it can be difficult to do, or you can look it up in a computer database. And to do that, there's a lot of different programs out there now. This is BLAST. You can find this at the National Library of Medicine. I just searched for something. I just copied and pasted it from somewhere. And you'll see that you get this kind of output, and it's looking at how well your sequence matched what you're doing. And then if you click here, it gives you the protein sequence, which is kind of nice. And the amino acid sequence is in a letter format, and it is going to give you can then use that to figure out the amino acid sequence. And I don't quite understand all of this, and then you can kind of backtrack potentially to the DNA. So there's a lot of genes that have been isolated and sequenced, and there are all sorts of things out there. Mine, I guess, was classified as unknown. And I did spend a few minutes trying to mess with this program and didn't get very far. A lot of what it is using, there we go. So here's a little bit on this protein. They give you just some information. It's probably not that exciting, but there you go. And you can just have it right there at your fingertips if you know what you're doing. It won't take long at all, but that can be hard. All right, so you find the target, and that target is going to also be kind of with your goal, right? What you're looking for, what you want to do and accomplish with that gene. All right, the second is really isolating that DNA. Right, we determined the DNA sequence if we have the protein and maybe using the database, but you need a physical copy of that DNA to do whatever you want to do with it, and how the method in which you want to insert it into an organism. And getting the isolation and getting this DNA technique or sequence, we're gonna skip. So I'm not quite as familiar with it, and I think when I was looking up, it seemed pretty complicated. But once you have that, you can copy it, and it's a pretty well-known technique now. It's called the polymerase chain reaction. And if you were listening to the governor talk about his testing strips, the 500,000 that he bought from South Korea, from what I heard, I didn't double-check this, so I might be wrong, but I believe they're using PCR, polymerase chain reaction, to amplify the DNA to see if it's the viral DNA or not. Supposedly it's going to be quicker if you have one of these machines. And it can be quick, right? Polymerase chain reaction is just a machine. All right, if you were to get this, you would be buying a machine, and that machine its goal would be to copy your DNA, and it can be what's revolutionary about this, and it's been around for a while now, is that you can, within a day or less than a day, you can have millions and millions of copies of DNA. Right? There are millions and millions of copies. And what this machine does, the simplified version of it, is you have a machine with all the DNA bases and the enzymes you need for DNA replication, the helicases, and your DNA polymerases. And you put your DNA that you isolated, maybe you have 1 or 2 copies of it from the lab, and when you put in this machine, and what it does is really just a heating cycle. The heating cycle kind of breaks the DNA, and then the enzyme's going to work and put the primers on, and then the DNA polymerases come through and they copy the DNA, and then you have, as you can see here, 2 copies. And you have this cool down and warm up, and as you do this, right, you continually make copies of DNA, and if you're doubling it very shortly, right, if you double it within a number of cycles, and these cycles, I believe that, and they're not that time intensive, and very quickly you have millions of copies of that DNA, and it's a technique that is used frequently now to just get copies. And once you have that copy of the DNA, that's kind of where the fun begins as far as genetic engineering goes, and what you can do with inserting that DNA, and what the flexibility it gives you to change organisms, and you can be that mad scientist. All right, I think that's enough for today. So I think the next one we're going to talk specifically about the techniques in which we can modify organisms once we have this DNA isolated. Enjoy.